There we go. Okay, so it's good that we start recording because I want to show you something that is relevant to the first exam. We have this slide on countercurrent heat exchange in aquatic organisms that's in the book. And I have to confess that it's a difficult thing to get across, partly because of the way it's presented in the book and in that diagram. It's just not clear exactly what the deal is with that. So I drew a little diagram that I think might help all of you, and it's now posted on the PowerPoints for exam one. And I want to run through it just very briefly before we start talking about populations. So the key thing about this business of countercurrent heat exchange, helping large aquatic organisms maintain body heat, is that the anatomy of the circulatory system is actually different. And I don't think the book adequately conveys that. And I've been trying to teach from that slide for a very long time, and just people are just not, they just kind of don't get it because they can't visualize how the anatomy differs. So I'm going to walk you through. This on the top is a regular aquatic organism. This is a fish that does not try to maintain an elevated body temperature. Fish have a two-chambered heart. So they have an atrium that collects blood from the body and pumps it into a ventricle, and that ventricle pumps it to the gills. And it's very simple organization. Okay, well, that deoxygenated blood has been circulating through the body. It's picked up body heat. What happens to it? It goes to the gills, and any heat that's in that blood gets lost to the surrounding water instantly. So this blood has little or no oxygen, but at least is warm until it gets to the gills. When it gets to the gills, all that body heat is lost. Then you've got oxygenated but cold blood going from the gills along the outside of the fish in these arteries, and then plunging into the muscles. Okay, in the muscles, it gives up its oxygen and absorbs the heat. Now it's cold blood going into muscles, so it's cooling the muscles. And then any of the heat that it does pick up is immediately lost in the gills. That's a big reason why fish in particular don't attempt to maintain a high body temperature. They can't afford it. There's no alternative for them but to expose their blood to the outside water if they want gas exchange. But in order to do that, they have to also accept that they will lose heat to that water. And also there'll be a diffusion gradient of ions that will cause them osmotic problems. So it's a necessary evil. They have to have gas exchange, and they have to accept that those other things are going to happen. There's no alternative. Okay, so the difference in anatomy in the fish that are big enough to maintain a body temperature higher than the water is this. Okay, the heart is still doing what it does. It pumps the blood to the gills. Okay, any heat that's in the blood gets lost to the surrounding water, but there's very little heat left in that blood. And here's why. So you got that oxygenated cold blood coming from the gills along the outside of the fish, just as in this fish up top. And then it plunges into the muscles and it gives up its oxygen and it gets warm. And then it is collected in veins that circulate not directly back to the heart, but to the outside of the fish, parallel to the incoming arteries. So this cold blood that's plunging into the muscles is right next to hot blood that's leaving the muscles. And so it absorbs the heat from the outgoing blood and carries it right back into the muscles. Basically, any heat that's generated by the muscles is not lost, it's transferred right back in. So the muscles can maintain a higher temperature. Okay, so once that happens, this deoxygenated blood then has lost all its heat. It's cold, and then it circulates back to the heart. And now the heart is pumping deoxygenated cold blood to the gills. So there's very little heat left to be lost. Most of that heat has stayed in the muscles right here. Okay, so it leaves the muscles here, but then goes right back in as the cold blood is warmed by countercurrent flow right next to the outgoing veins. I think that this will help you visualize how this actually works. I've been very frustrated by trying to teach this for a number of years, and it just hit me, you know what, I just need to draw the anatomy. So I do hope that helps you. It's an important issue, and it's one that's really ingenious. It's a totally ingenious adaptation, altering the anatomy of the circulatory system to conserve heat. And I hope this is a better explanation of it. Okay, now for the topic of the day. Where are we here? There. We're going to move now from talking about adaptation to talking about the biology of populations. So we're going to take a conceptual jump here. And instead of talking about individuals and their characteristics and how they live in particular areas, we're now going to focus our attention on groups of individuals and the characteristics of groups of individuals. Remember, there will be emergent properties. Some of them are listed on this slide. So a population is a group of individuals of a single species inhabiting a specific area at a given time. Okay, and one of the factors that we generally think of when we try to define a population is, could they potentially interbreed with each other, right? So if you have grizzly bears in Alaska, are they the same population as grizzly bears in Montana? Well, you know, there's like 3,000 miles in between those two places. A lot of people would say, well, it's not really fair to call that one population because the ones in Alaska and Montana don't interbreed. Not that they can't, but they're just physically not able to find each other. So that's why we distinguish demes, which are subpopulations. Okay, so that's the distinction. You can define a population as big or small, but you may want to subdivide it into demes that actually have the ability to interbreed. Okay, so the 6 characteristics we're going to focus on in terms of populations are as follows. Today we'll do the distribution and dispersion. So distribution is the occurrence of a population on a geographic scale. Dispersion is the association among individuals on a local scale. And then in the next lecture after the exam, we'll talk about abundance and density. Abundance is the commonness or rarity of species, and the density is the number of individuals per unit area. So abundance has more to do with kind of global numbers, whereas density is a local thing. Then we will spend most of the next couple of weeks talking about the following 2: age distribution, which is the relative number of individuals of different ages that you find in a population, and how that affects the dynamics of a population. Like if you have a lot of young individuals, what does that do for population growth or decline or persistence? And from that, we will then derive growth rates. And that will tell us whether populations are growing, declining, or stable. And we will spend most of our time on that last point. But realize that all of these are emergent properties of populations. Okay, so today our focus is on the first 2, distribution and dispersion. Now, the distribution of a species depends on its characteristics. And the characteristics of a species define what its needs and tolerances are. And we call that kind of abstract collection of information the niche. The niche is not a place. The niche is kind of your shopping list. It's the environmental factors required for survival and reproduction of the species. And you can't just survive, you also have to be able to reproduce. Now, we think of the niche in a couple of ways. First, there's the fundamental niche, and that's the theoretical collection of all needs and tolerances under which a species could potentially survive. However, organisms have to share the environment with other species in any real community. And so they don't usually get to occupy their entire fundamental niche. They usually get to occupy only a part of it. So the realized niche is the set of conditions that a species can actually occupy in a given community based on its competition and predation relationships with other species. So the niche is your shopping list. The habitat's your grocery store. It's where you actually find those things. The habitat is the physical location where the niche requirements are fulfilled. So the habitat's a place. The niche is not a place. The niche is an idea. So the geographic range of a species in broad terms is defined by the realized niche. Okay. And although the fundamental niche may potentially define where the species is, we have to take into account that there may be areas on the face of the earth that include the fundamental niche where this species doesn't occur because of competition or predation. So yeah, fundamental niche basically tells you what conditions to look for to find a species, but where you actually find it is kind of the realized niche. So the geographical range is the area on Earth where you find that population. Now, it's important to understand that within that area, that doesn't say that the entire area is suitable for that species. It's just the broad outlines. There may be patches within that area, and this slide attempts to kind of illustrate the patchy nature of niche conditions. So here is a rare plant, Fremont's leather flower. Okay, and it's restricted to 3 counties in Missouri, but its niche is defined by dry, rocky soils on limestone outcroppings. Well, these 3 counties in Missouri are not composed strictly of that set of conditions. There are patches within those 3 counties that have those conditions. So here is an overall map of the 3 counties, and the little green things show you the distribution of these limestone glades where the plant occurs. So the geographical range may be these 3 counties, but that doesn't mean it occurs everywhere. The niche conditions are actually only fulfilled in very restricted areas. So we're going to zoom in further and further, and here is a little stream course, and you can see that there are patches of these plants along these streams. So the glades are distributed along stream beds, but the hills and the stream valleys don't have those glades. So here's where you actually find the niche conditions being fulfilled. And so here at the very most detailed level is a map of individual plants and their spatial relationships to one another. So it's really important to understand that although you might have these glades available, if there's a herbivore that eats this plant, it might consume all the plants in one of these little patches. And so just because the niche conditions are there, it might not be part of the realized niche. Okay. Now, it's really important to try to understand whether species are limited by their niche, by the conditions that they require, or by interactions with other species. And so we frequently do the experiments in which we try to separate those things. So here's an experiment where we have 2 kinds of monkey flowers, and we have this Lewis's monkey flower, the kind of pinky one, That grows at relatively high elevations that are defined by this gray bar. The scarlet monkey flower has this kind of salmon colored bar, and it grows at much lower elevations that are kind of obscured by this little bar at the bottom of our graph. Sorry about that. But we can ask, well, is Lewis's monkey flower out competing the scarlet monkey flower at high elevations? So are these flowers limited by their niche, their fundamental niche, or are they limited by interactions with other species? So just because Lewis's monkey flower occurs at high elevations doesn't mean that that's its fundamental niche. It might be able to occur everywhere in the absence of scarlet monkey flower. So let's grow them together in gardens at different elevations and see how they do. So when they're planted outside their natural elevations, they grew poorly. So here's the proportion surviving at different elevations. And you can see that Lewis's monkey flower survives well at high elevations and really badly at low elevations. Interestingly, the scarlet monkey flower does not so bad at high elevations. However, look at the stem growth. Okay, so it can survive, but it can't grow. So basically these plants, you can plant them there, but they're not going to be able to grow and they're not going to be able to reproduce. So in fact, these two plants are in fact limited by their fundamental niches. They are not competing with each other so much as they are limited by their niches. Now, if we could show that the two species were equally able to survive at all these elevations and grow at all these elevations, we might have evidence that they're competing with each other. So it's an important point in ecology that we try to understand what does limit these populations. Okay, so that's a little tour of the concept of distribution and what limits distribution. This is a relatively short presentation. So now we're going to move to the second feature of populations, and that's dispersion. That's the association of individuals on small scales. Now, there are three kind of archetypal patterns of dispersion, and they tell us really interesting things about populations, really almost just by counting individuals and asking how close are they to each other. You can get a surprising amount of information about this. So that's why we do these kinds of studies. So dispersion can be random. And in a random dispersion pattern, there's an equal chance that individuals are anywhere in the environment. Okay, usually that suggests that the resources and environmental tolerances that these organisms have are kind of uniformly distributed. And there's sort of no interactions among these individuals. Okay, if the dispersion pattern is regular, then we find individuals uniformly spaced. And that's really interesting because it implies a couple of things. It implies that individuals may have the exclusive use of an area, that they avoid one another. Why might that happen? Well, maybe there is a limited amount of resource in a given area and it can only support one individual. So two individuals cannot be too close to one another. Okay, so that implies that competition may be happening. The third pattern is clumped or aggregated, and in clumped or aggregated patterns, there's a large amount of space where nobody occurs. And then there are a few patches where pretty much everybody occurs. So that could be due to mutual attraction between individuals. For instance, if you need to find a mate, or it may be because that your resources are very patchily distributed and you just have to all congregate to get those resources. Now, this is the subject of one of our labs. And so if you've been in lab, you'll know that we did measure dispersion patterns in some trees on campus. And we used a particular method to do that. I'm going to at least explain a little bit about the method, but we're going to talk about a second method as well. So here's an illustration of dispersion patterns. So, and what is this? Make that go away. I don't know what that is. People are very bad at drawing random dispersion patterns. They almost always draw regular patterns. But here's an example of a random pattern that was actually measured in nature and kind of transcribed into a cartoon. So this is random. It implies neutral interactions and between individuals and between individuals in their environment. Regular patterns are evenly spaced, and it implies that organisms are antagonistic, they're competing for something, or that resources are easily depleted. And then finally, we have these clumped or aggregated patterns, and it implies something that about there being attraction between individuals, or maybe resources not being equally dispersed in the environment. So this is often the basis of the study of the relationship of organisms in their environment. And it generates lots of hypotheses. Like if you see a regular pattern, you might then hypothesize, well, competition is probably generating that. But these dispersion patterns are relatively easy to measure. Here's an example of real world dispersion patterns that I had a hand in measuring. So these are 3 species of plants in the rainforest of Barrow, Colorado Island in Panama. And my first job as a graduate student was to go down to Panama with my major advisor and survey, that is to lay out a plot in the rainforest where we measured the location of every single plant, more than 250,000 plants. So that was really cool, and it formed the basis of a whole network of such studies now around the world. There's at least 20 of these plots, but this is the original forest dynamics plot. So after we mapped the plants, this is what we found. We found that some of them are kind of regularly dispersed. And you can see that it's not that everybody is exactly close to each other, or exactly the same distance from each other, but the relative distance between individuals is not that different. Okay, and these are animal dispersed. Okay, and that's because the reason that you have this dispersion pattern is because animals tend to avoid one another, and they poop out these seeds pretty much within their little their territories, and they defend their territories, so they go all around their territories, and so they disperse them pretty regularly. Here is a second plant that is dispersed. It's a big tree, and so it's dispersed by having their seeds blow in the wind with these little wings. And so you see that this is much more random. And some plants are extremely close to one another, but others are very far away from others. And this pattern may not look all that different from this one, but when you measure the distances among individuals or the number of individuals in an area, you see that there's really kind of no consistent pattern compared to this one. Finally, here's one. This is a little shrub that is ballistically dispersed. Okay, so what that means is that these things form a seed in a little pod, and the pod dries out from the base towards the tip and basically pushes out the seed, and as if it was a pea shooter, and sort of flips it a small distance away from the parent plant. So it's aggregated around the parent plants. So you don't find them out here because there are no parent plants out here, and they can only be dispersed a couple of meters at the most. So there's a real life aggregated pattern. Here is dispersion patterns in aggressive and non-aggressive stingless bees. This is another study I was involved in. So we have two species of bees. This is Trigona fulviventris. Trigona fulviventris is a very common bee in tropical rainforest, and they're very aggressive. And they have basically wars between colonies. And so the colonies are regularly dispersed in the environment in this patch of forest. And they fight with each other, and so they cannot afford to get too close. Trigona dorsalis is another lovely stingless bee, and they are very unaggressive, really nice bees. As the name implies, they're not a menace to people. But you see that some of them are kind of distant from other colonies, and others are very close, and that's because they don't really interact. Okay, so the important thing to know about dispersion patterns is that they're scale dependent. At small scales, any species may show random, regular clump distributions depending on the local conditions. But at large geographic scales, remember we have the geographic distribution pattern. Well, that's going to be, by definition, a patch on the face of the Earth. And there'll be a huge amount of the face of the Earth that is not occupied by that species. So at large geographic scales, all species are clumped or aggregated. That's because the niche conditions don't occur evenly across the face of the Earth. So small scales, you get these different patterns. At large scales, every single species you can imagine, maybe except people, is aggregated. So here's two examples. Here's crows, and this is the American crow, and you see there's high density of American crows in particular areas. And in some parts of the country, like the arid grasslands, you don't get American crows at all. Down here on the bottom, partly obscured by our bar, is the distribution of fish crows. And fish crows are found mostly along the Gulf Coast. And I'm sorry, you'll have to see that Louisiana is kind of a hot spot for fish crows. But it's clearly an aggregated pattern. Now, if you go into any of these areas where they do occur, you may get regular, random, or clumped dispersion. So let's finish up talking about one other thing. Dispersion patterns are frequently time dependent. Here's a really great example: creosote bush in the western deserts. Adult creosote bushes, as you see here, are pretty regular. And that's because they are limited by moisture. Okay, the root zone of any individual creosote bush is pretty much exclusive. The amount of moisture in the area surrounding a bush is sufficient to only support one adult bush. And if they grow too close together, one of them is going to die. So it's a regular dispersion pattern. However, when they reproduce, the young plants tend to grow pretty close to the adults, and it's aggregated or clumped. You get an adult surrounded by many juveniles. Okay, and so that's an aggregated pattern. But then some of those plants are going to start to die for lack of water. And that's going to change this aggregated pattern into more of a random pattern, because which organisms die is going to be kind of random. And so in the end, you'll get this regular distribution of the survivors. So dispersion patterns are scale dependent and they are time dependent. Now, talking about measuring dispersion, two methods. First, you can measure the distance between nearest neighbors in a defined area. This is what the laboratories did. Okay, so what you do is you compare the expected distance between nearest neighbors, just based on the size of the area and the number of individuals in the area, to the actual distribution that you saw. If plants or animals are significantly closer than you expect to find them, they're aggregated. If they're significantly further than expected, then it's regular. Okay, so the thing is that these are kind of complicated calculations and they're based on a t-test and they're hard to explain to a class where you're just showing slides. So I'm not going to talk much about that. This is what the lab did. This is the method the lab used for their project this semester. A really easy thing to talk about, though, is a second method in which you count the number of individuals in a series of plots. Okay, they all have to be the same size, but count the number of individuals in those plots. Then calculate the average number in each plot and calculate the statistical variance of the mean. So the variance is related to the standard deviation. It's a measure of how widespread the observations are around that mean. Okay, if you calculate these two things and you divide the variance by the mean, the variance to mean ratio, as we call it, is a really intuitive little rule of thumb, and it's really easy to explain. So if the variance is about equal to the mean, that's a random distribution or a dispersion pattern. If it's much greater than the mean, then high variance means that the numbers are all over the place. So some plots will have 0 and others will have many. Well, that's aggregated. And if the variance is much lower than the mean, it means that all of the plots have about equal numbers. And that's a regular dispersion pattern. So you test that by comparing to a random dispersion pattern that you generate using what we call the Poisson distribution. You just use the mean of the number of individuals in a plot and plug it into this distribution generator, and it gives you some numbers that you can test as an expected value. So if you have 100 plots and you see that there are 20 plots with 0 individuals, you can use the Poisson distribution to ask, how many do you expect to have 0? And then you can use a chi-square test to calculate the difference. So we're not going to do that. We're just going to look at how the variance to mean ratio works. And here's an example that illustrates kind of the random, aggregated, and regular dispersion patterns. So here's the random at the top. Okay, and if the number of individuals in a series of plots is not different from what you expect in a random distribution, Poisson distribution, then it's random. If it differs significantly from the Poisson distribution by a chi-square test, then it could be clumped. And here you see you've got a 0 and a 0 and a 0 and a 0 and a 0. So you got 5 zeros in here. And you got several plots that have multiples. And so here's one that has 6. Here's one that has 4. Okay, so that's more than you expect. And that's a clump distribution. And down here at the bottom, pretty much everybody has the same number. It's either 1 or 2. There are no zeros, and that's regular. There are fewer zeros and large numbers than you expect. So I'll give you an example of using the variance and mean to figure out this ratio. So here's those plots, and here's what we did. We counted up then the number of organisms in each, and the mean is 1.56, the variance is 1.46. All right, well, 1.4 divided by 1.5 is very nearly 1. And so it's about equal. And so this is a pretty easy to distinguish random dispersion pattern. Okay, over down here in the second one where we had 5 zeros and we had a 6 and a 4, the mean is 1.8, the variance is 3.1. That's a lot larger than 1. So divide 3.1 divided by 1.8, you get 1.7. That's way larger than 1. Okay, that's an aggregated or clumped dispersion pattern. And of course, you can use the chi-square test to actually test the significance of this difference from random based on the observed numbers of 0, 1, 2, 3 individuals in a plot. We're just looking at the variance to mean ratio here. Here's the regular dispersion pattern, and the mean is, once again, this is a mean of 1.18. The variance, though, is really small, 0.16. And although you can't see it, if you divide the variance by the mean, the variance to mean ratio is 0.13, so almost zero. So there's very little variation in the numbers per plot. And that means it's a regular dispersion pattern. So I don't care if you understand how to statistically test these things for the lecture. That's something that our labs did and had a little more time to work that through. What I want you to know is what a random, a clumped or aggregated or regular dispersion pattern is, what it implies, and how you would detect it using the variance to mean ratio. I'm not going to generally ask you to calculate the mean or the variance, but if I gave you a variance to mean ratio, could you interpret it for me? So that is something that we will cover on the second exam. And for now, we are kind of done with today's presentation. So we have a few minutes here. And if you would like to ask a question, I'd be happy to answer it. Or it doesn't have to be on this one, it can be on anything else. Okay.
I have a question.
All right, Lauren.
About the sorry, did you say Lauren? I didn't hear you.
I'm sorry, what was that?
I'm sorry, I thought you said something. I think my connection is kind of bad right here. I have a question about the ocean's acidity. Are our oceans like really acid, like acidic because of the bicarbonate, the water and the bicarbonate or whatever? With like the excess of that would be an extra like protons, right?
Right.
So does that mean our oceans are acidic?
Okay. Ocean water is currently slightly on the acidic side. However, most of the CO2 that goes into the atmosphere from burning fossil fuels is absorbed by the oceans. And so
Right.
the oceans are acidifying very rapidly. And they are acidifying to the point where they will exterminate all aquatic life very soon. So this is one of the major concerns that I have about continuing on our course of burning fossil fuels. The mass extinctions of the past have all pretty much been accompanied by ocean acidification and mass extinction of aquatic organisms, maybe not due to temperature change in the ocean so much as interference with metabolism. So one of the benefits of having oceans is that we do not experience the full impact of climate change because the oceans are our buffer.
Yeah.
So it's like turning on a gas burner on a stove. If you put your hand directly on that gas burner, it will burn you. But if you put a pot of water on that gas burner and put your hand in the water, well, the output of the burner is the same. But the water is absorbing that heat and it has a high heat capacity. So until the water absorbs all the heat, you're not going to feel the effects. That's the fool's paradise we are living in right now.
Yeah. So like, I don't know. Is there nothing that, I mean, I guess there's no way of fixing it. Is there any way to like get the acid out of the water if like carbon dioxide was lowered?
Okay. Yeah.
Or, like adding chlorine?
Well, there's an equilibration between the atmosphere and the oceans. If you stop putting CO2 into the air, it will stop diffusing into the oceans. And if you start, say, regrowing forests, or maybe using active carbon capture technology to remove CO2 from the atmosphere, which is feasible, which the Swiss are already doing in big plants in Switzerland. I think there's one being built in Sweden. There is absolutely no reason why we couldn't do that. Then that will draw CO2 out of the oceans, back into the atmosphere, but at lower levels, of course.
Okay. So, I mean, it would take time, but cool.
It would take time. You have to understand that we are living in a world that has what scientists term hysteresis, that is a delayed impact of a change. So the carbon that we put in the atmosphere today will not reach its full impact on the climate for some decades to come. That means that you will live your entire lives with a warming climate. And the way to deal with that is to stop putting carbon into the atmosphere. And I think that we will also have to start actively bleeding CO2 out of the atmosphere. It's much more efficient to remove CO2 from the atmosphere than to remove it from water. So that's probably our most efficient bet. Just start removing CO2 through the growth of plants and maybe through active carbon capture technology that will gradually pull CO2 out of the oceans. And we will eventually succeed in reducing carbon levels to tolerable levels. But it's not something that's going to happen by 2030.
No, I agree.
Something that will hopefully start having an effect between 2050 and 2080.
That's crazy to
I hope you live to see the peak and the decline.
So another question about it. Sorry, would like kelp be able, does kelp use CO2 the same way as other plants do? Or they're in symbiosis, or they're symbiotic?
Oh, yeah.
I don't remember.
Yeah, kelp is a huge photosynthesizer.
Okay.
The problem with kelp is that it doesn't sequester that carbon in, say, woody structures. They don't make wood. So when a kelp leaf dies, it just decays within a couple of days, releasing all that carbon back. That's kind of the problem with aquatic plants. Terrestrial plants that make wood out of lignin and cellulose, they sequester carbon for a much, much, much longer time.
Well, like, aren't kelp farms very popular? I mean, would it be like, it just would be unrealistic to have really big kelp farms and just like, I guess, just scoop them up and get rid of them and start them up again to remove?
Well, that would certainly help. I mean, if you could easily harvest tons of kelp per acre of ocean surface every single day, and it would regrow almost immediately. But then you have to figure out how you're going to dispose of that, and,
Right.
prevent it from just being released in the atmosphere very quickly.
Okay. Okay. Thank you.
Okay. Other questions about our predicament or about the exam?
Hey, I have another question. I feel like I'm confused. I'm a little bit confused on how the air circulation shifts seasonally, and like, I was looking at the exam one, the question about like the winds in New Orleans when it's 30 degrees north.
Mhm.
And I'm just a little bit confused. Is there any way you can explain to me like how the air circulation shifts like seasonally, and like what position would we be in? And I don't know, I'm just a little bit confused on that.
Okay, well, recognize that 30 degrees is about the break point between easterly winds and westerly winds, right? You've got falling air masses, and they tend to fall at about 30 degrees north or south, and then they either go back towards the equator or towards the poles. So New Orleans is directly under that column of falling air. That's how it is when the sun is heating the actual geographic equator at its greatest rate. But in the northern summer, the northern hemisphere is closer to the sun, and the geographic equator is no longer the solar equator. The solar equator is pulled north. Okay, well, that means that the column of falling air is also pulled north of New Orleans. And so the wind that hits the ground north of New Orleans is going to flow back towards the solar equator. From which direction? The east. In the northern winter, the solar equator is pulled way south, And so the Hadley cell is pulled way south, and that column of falling air is also pulled way south. And that means that the New Orleans area is now not directly under the equatorial Hadley cell, it's under the mid-latitude circulation cell, right? The Ferrel cell. The winds under the Ferrel cell are blowing from west to east. So during the winter, we have west winds from the Pacific. As the seasons change and the solar equator is dragged north, we fall under the Hadley cell where the winds are blowing from the east. Winter, Hadley cell moves south, Ferrel cell moves over New Orleans, winds are from the west.
And the winds will be coming down instead of 30 degrees, it would be coming down at 60 degrees.
No, it rises at 60 degrees.
Why is that 60? Okay. Thank you.
Okay, I think we have time for maybe one more. I do have to go. I'm trying to get somebody to tarp my roof. Anybody? Okay, well, you can email me from now until the exam. Recognize that the exam will be on Monday at 11 unless you've made a prior arrangement. And don't hesitate to email if you do have a question. I will be giving you a little more information about how how the exam's going to actually work, and how you'll log in and how you'll be prompted to do the lockdown browser. So, be on the lookout for that. And hopefully we'll see you back in class on Wednesday. All right. Goodbye. Have a great afternoon.
Thank you.
You.
Thank you. Have a good
Have a
one.
good weekend.